Hi, this is Mr. McLean. In this session, we're going to be taking a look at section 9.2, which lets us examine the process of glycolysis, and it allows us to focus in on these two components, which is the investment that is required to initiate glycolysis and the payoff we get from glycolysis. Now, before we get into that fully, what I want to do is I want to take a look at this particular slide. I didn't have time in my previous video to get to this, but I think it's important because it kind of shows us the multiple steps that are occurring in the overall process of cellular respiration. So if we start over here with glycolysis, one of the things that we notice is this is occurring in the cytosol. And what that tells us is this process can occur in both prokaryotes and eukaryotes. In fact, it is what we refer to as an evolutionary relic. It's something that existed billions of years ago in some of our earliest cells, and of course it exists today when we start to think about our own cells that initiate cellular respiration through glycolysis. So basically, what we're going to find is we're going to have an investment of glucose and some other things into the glycolysis process. And in the process of breaking down through redox the glucose, we are going to be able to produce some NADH, and that's because of redox. And that's going to carry with it electrons and hydrogens. We're also going to produce a little bit of ATP, which we're going to see is going to be equal to 2 molecules. Put that right there, 2 molecules of ATP. That's coming through what's called substrate level phosphorylation. I'm going to go over that here in a minute, but that's important to know that mechanism that's at play. And most of the energy we're going to see that comes out of glycolysis is still untapped in the form of pyruvate. So if we're dealing with a prokaryote, that kind of ends the progression. What we'll have to do is we'll have to take pyruvate and we'll have to break that down using some other mechanism, anaerobic respiration or fermentation. However, if we have a situation where we do have a functional mitochondrion and we have oxygen, then we'll proceed into the next couple steps. So one of the things that we're going to see is that pyruvate is going to be actively pushed into the mitochondrion, and it's going to do what's called the conversion of pyruvate. So we'll go over the details of that particular step. And then the conversion of pyruvate is going to prepare for us acetyl coenzyme A, which is going to dump into the citric acid cycle. Citric acid cycle is also known as Krebs. It's also known as TCA or tricarboxylic acid, and that is occurring in the matrix. Okay, so that's this inner area of the mitochondria. And from that, we're going to make some ATP, but only 2 molecules. And once again, substrate level phosphorylation. Okay, we're going to produce, coming off of that, a certain quantity, which we'll detail in a few minutes, of NADH and FADH2. And once again, those high energy molecules are going to deliver their electrons to the final component, which is known as oxidative phosphorylation, which involves the electron transport chain. And the electron transport chain is like located right here. Okay, so if you think about our mitochondria with the multiple folds and the high amount of surface area, that's where the electron transport chain is going to be found at. And once we do our oxidative phosphorylation and chemiosmotic ATP production, we're going to be able to make a quantity of energy that is far outweighing the previous steps. And it makes it through what's called oxidative phosphorylation. So right now, as of the time of publication of this video, the accepted upper limit of ATP production from one molecule of glucose is a total ATP equal to 32 molecules. And that's from one glucose. So for every one glucose molecule, you get 32 ATP molecules. So that means that 28 of those are coming from oxidative phosphorylation. So it's quite important for you to remember that if you come across any questions about where's the majority of the ATP being produced, know that the majority of it is produced through the oxidative phosphorylation process, which we will spend a significant amount of time on. Now, just real quickly, you saw me mention the term substrate level phosphorylation. The big difference between oxidative and substrate level phosphorylation is where the phosphate's coming from. So if it's substrate level phosphorylation, what we're finding is a substrate is going to go through there, and it's going to be the source of our phosphate. So we have some substrate with a phosphate. There's an enzyme that's going to carry out the reaction, and we're going to be able to transfer that phosphate to the ADP. Once that happens, we get a product that no longer fits in the active site, so that'll be ejected out of there, and we get the production of ATP coming off of it. So if you compare substrate level and oxidative phosphorylation, substrate level makes only a small fraction. 90% of the ATP is generated through oxidative phosphorylation. Okay? And that's important for us to know. So all of the ATP that's produced through glycolysis or the Krebs/citric acid is substrate level phosphorylation. The remaining, the majority of it, is through oxidative phosphorylation. Okay? So I'm not going to spend a whole lot of time going through all the steps of the glycolysis pathway. I have attached in your Google Classroom a detailed sheet, and of course we did it in class. So I'm going to highlight the major portions of it and try to emphasize what's going in and what's coming out. So overall, if you look at the investment component of glycolysis, we're going to invest a glucose molecule. So that's our organic source. We're going to invest 2 molecules of ATP. We're going to talk about what they're going to do, but basically it's going to destabilize the molecule. We're going to have a payoff stage, which is actually going to form 4 molecules of ATP. It's going to form 2 molecules of NADH, and it's going to form 2 pyruvate molecules. So from a net standpoint, we get 2 pyruvate plus some water. We net 2 ATP because when you subtract out what you've invested, you have a 2 remaining, and we net 2 NADHs. All right, so let's take a look at the process. So going through glycolysis, you start off with your glucose molecule, and right now you're going to notice that the first investment of ATP phosphorylates that glucose. So we refer to this new molecule, didn't mean to do that. We refer to this new molecule as glucose 6-phosphate. And the reason why is we've attached a phosphate onto the 6th position of carbon in that molecule. Okay, and that's a step towards destabilizing the overall molecule. All right. Now, if we move on to our next step, our glucose 6-phosphate, we're going to see that it's going to go through a change. So in our diagram, I showed you the conversion from something being glucose to being fructose, and basically that's an isomerization reaction. Okay, so it's still going to have the same number of phosphate groups, which is 1 at this point. It's still going to have the same chemical ratio. It's just arranged in a different isomer. Okay, the next step, we're going to come back to this again, but this is one of the regulatory enzymes I do want you to know. That is the PFK, which is phosphofructokinase. That's the enzyme that controls this second phosphorylation. So we've already invested 1 ATP to give us this first phosphate. The second investment fully destabilizes this molecule. It now becomes fructose 1,6-biphosphate because there's a phosphate in the first and the sixth position, and it makes that molecule highly unstable. The thing about it is that phosphofructokinase is allosterically regulated in a variety of different ways. One of the things that can act as an inhibitor of PFK is actually ATP. So if you think about it, we run glycolysis as an intermediate step to produce ATP. When our ATP reserves build up, the cell tries to remain efficient. It doesn't try to stockpile. And essentially the accumulating level of ATP as well as citric acid will actually inhibit the PFK enzyme and thereby stopping the process. When the ATP levels are low, this process will actually proceed forward. And I'm going to show you at the end of the chapter that there's actually some things that will boost this particular process when the levels are elevated, such as adenosine monophosphate. All right, so assuming we made it through that step, now we have this unstable molecule, and the unstable molecule is going to split into 2 3-carbon molecules. So let's address this side first. This molecule we refer to as G3P. It's glyceraldehyde 3-phosphate. This is the sugar that we want. It's a 3-carbon molecule, and basically as soon as it's formed, it continues on with the process. There is an isomer of G3P known as dihydroxyacetone phosphate. There's an isomeric reaction or an isomerization that takes place between these two molecules. So basically, when one G3P is made, it automatically proceeds to the next step. When the dihydroxyacetone phosphate is made, it must go through this reaction where it converts into G3P. As soon as it becomes G3P, it proceeds, it continues on with the reaction. So the idea is this is a reversible reaction that never really reaches equilibrium, because as soon as it gets to G3P, it continues on to the payoff stage. All right, so now we're on to what would be considered the back half of our diagram that we did. This is the payoff. So the first half was investment. Now we're talking about getting some energy payoff. So keep in mind that everything that we see now is actually doubled, because when we left off here, we were going to break that 6-carbon molecule into 2 3-carbon molecules. All right, so we're going to follow one of those 3-carbon molecules. So here's a G3P. First thing that we notice is redox. Okay, so this is showing 2 because it's saying that there are 2 of these. That's why it's showing that. There's one that's happening to this particular G3P, but there's actually 2 G3Ps. Just keeping that in mind. All right, as that happens, it picks up some phosphate from the cytosol. So once again, we've got a 3-carbon molecule, but it has 2 phosphates on it. As we go through here, we start to produce ATP. All right, one on each side. We're going to produce a molecule of ATP coming off of this one. We're going to produce a molecule of ATP coming off of the matched pair. As we go through here, we get a variety of different rearrangements and changes. Not totally necessary for us to memorize those different things, but as we're progressing towards the bottom of the Krebs cycle diagram, one of the things that we see is water is given off. So we release water off of each one of those side chains, and as we get down to the very end, we make an additional pair of ATP. And the final molecule at the end of the Krebs cycle, which holds the majority of the energy, is pyruvate. Pyruvate has not yet been broken down, and the rest of our energy reserves should be coming from pyruvate. Okay. So hopefully that helps.